So, I welcome you to an entire new chapter. And this new chapter that we have got, so this is entitled as N enzymes. Now, the situation that we have to consider is that why is there need or the necessity of the enzymes for the normal working of the cells or the normal working of the body? Now, the slight issue with our body is that the temperature of our body, this is kept nearly 38 to 39 degrees centigrade, and this is such a low temperature, and in this much low temperature, the chemical reactions they cannot be carried on at a very high speed. Like if I were to tell you about some of the chemical reactions that happens in the cell, so you see from the previous lectures that what I have got here at this point, so this is the mitochondria, and in this mitochondria, the glucose and the oxygen gas they have to combine with one another to be releasing energy out of it. Likewise, if I were to say that this is the cell of the liver, so we have to break down the excess amino acids to be making the urea out of it. So these chemical reaction, if they were to be left at such a low temperature of the body, so our chemical reaction would be very slow. So we need to have something in our body that must allow the chemical reactions to be proceeding at a faster rate and that too at a lower temperature. Now, since the topic is enzymes, so we must know that what these enzymes actually are. So, you know that these enzymes, they are the biological catalyst. The first thing that the examiner is gonna ask you that what catalysts are. So, you say that the catalysts, they are the chemical compounds that would increase the rate of reaction without itself being getting changed. Now, the same kind of a thing this since this happens to exist within our living bodies, so that is why we entitle them or we term them as the biological catalysts. So the role of this biological catalyst would be that they would increase the rate of the chemical reaction, and by the rate what we mean is that in given time how much of the reactants that we have got they would be converted into the products. Now there comes another thing that these enzymes, how come they increase the rate of the chemical reaction? Simply by lowering the activation energy, and I will tell you in a while that what activation energy we mean by that. And these enzymes, they have another property that whenever they would be catalyzing some chemical reaction, so they themselves would remain unchanged. Like any of the chemical reaction they work upon or any of the reactant they work upon. So at the end of that chemical reaction, we will get our enzymes intact. So let us just try to see that what is the meaning of this word activation energy. Now you see as that wherever the chemical reactions are taking place, so there are two things that we have to be bothered about. That we will have these reactants, and these reactants they have to convert into the products. So whenever the reactants they have to convert into the products, so there is some kind of a barrier which these reactant particles have to cross. And if they would not be able to cross that energy barrier, so ultimately we will not have the products out of it. So this barrier which the reactant particles have to cross, so we call this as an activation energy. Now, for the time being, if I were to say that there is some specific chemical reaction, I have the reactants and I have the product, and this barrier of the activation energy basically implies that if the reactant particle have energy equivalent to 100 or greater than 100, then I am going to allow them to be changed into the product, or else I would not. Now you consider as that I have got multiple particles written here. Like this is the first particle having energy of 100. The second reactant particle have an energy of 102. The third one has got it as 99. And the fourth one has got it as 98. So if I were to assume this barrier of energy, so among these four reactant particles, I would either be able to convert this reactant into product and this reactant into the product. The reactants that have the energy lower than this activation energy, they would not be able to be changing into the product. So the situation or the scenario is that how come I be able to convert these reactants with the low energy to be reverting into the product? So there are two ways. Either I start to heat these reactant particles so that their energy could be released, or the other option is that I should increase this barrier of the activation energy. Now, for the living organisms, Heating our body is not a feasible approach to be booming or to be fastening the chemical reaction, but the most feasible approach is that we should be lowering this activation energy. And by lowering of the activation energy, we would fasten up the chemical reaction. And in our body, we have got the enzymes that would lower the activation energy. So the reactant with the less energy, they can also be changed into their respective products. So I hope now you would be clear that these enzymes being the biological catalyst, they increase the rate of chemical reaction. How? By lowering the activation energy, and by the end of the chemical reaction, they would themselves be not changed. Now, the other situation is that we must be very much clear about the situation that what are the characteristics and the properties that we have to remember for the enzymes molecule. The first thing is that what composition actually we have got for the enzymes. So we say that these enzymes they are made up of the proteins. Now you see that if I were required to be making an enzyme within my body, so I would take the amino acids. These amino acids, I would link them with one another, and then we would make polypeptide chains out of it. And these polypeptide chains, when they would be hold in a three-dimensional configuration, so as a result of that, I would be making the protein out of it. So simply, whenever the enzymes have to be made, so we need to bring about the linking of the amino acid, and by that we would end up making a protein out of it. Now, since the enzymes they are since the biological catalyst, so we must know that what are the factors that could have an impact on the working of the enzymes molecules. So among that, we have got total of four factors, but for your course, you just need to remember that this is temperature. Then we have this pH. The third is amount or the concentration that we would have for the number or for how much of the enzyme molecules we have got. And the fourth, you could say, the factor that would have an impact on the enzyme's activity. So this is the amount or the concentration that we have got for the substrate molecule. Now, when I'm saying the substrate molecule, so simply this substrate is a particle on which this enzyme would be working. The other scenario is that the enzymes, whenever we want them to be catalyzing some chemical reaction, so they are not required in large amount, but rather than we require them in very small amount. So a very small amount of the enzyme would be able to catalyze a large amount of the reactants. And this again is one of the very important benefit for our body that these enzymes, whenever they are contributing their role in any of the chemical reaction, so they would remain unchanged. Now, the other situation is that these enzymes that we have got, they are very specific. And by the word specific, what we mean is that for every reactant on which the enzymes have to work, so every enzyme would be special. Like if I would like the enzyme to be breaking down the sugars, so I would be requiring these amylases enzymes. If I were required to be breaking down the proteins, then I would require this proteases enzymes. And if I were to break down the lipids or the fats, so for that I would be in the need of these lipases enzymes. Now, before we proceed next, there were technical terms we need to remember in context of the enzymes. Here, I must take you to a situation that how many type of the enzymes we have got. So depending upon that whether the enzymes they are released within a cell or they are released out of the cells. So these enzymes they are further categorized into two of the distinct categories. And for the first enzymes that we have got, they are referred to as intracellular enzymes. And the second category of the enzymes that we have to be knowing as they are termed as or they are entitled as an extracellular enzymes we say. Now from the words you must be able to elaborate that the intracellular enzymes they would be produced within a cell. And whenever we will have extracellular enzymes, so they would be released out of the cells. So on the basis of where they are being released, like if they are there released within a cell, this is an intracellular. And if these enzymes, if they would be released out of the cell, so they would be the extracellular enzymes. So I would like you to quickly just see this, that intracellular enzymes, they are released within a cell. Like for instance, if I had this mitochondria and I was in the need of combining the glucose with the oxygen gas and I would require some enzymes So these enzymes, they would be working within the mitochondria, within the cell. Okay, and how I would be making an intracellular enzyme simply. The genetic information for the enzyme making would be available on the DNA. I would copy it on the messenger RNA. The messenger RNA will leave the nucleus and then it would come on toward the ribosomes. And then on the ribosome, I would link the amino acids. I would make the polypeptide chains and I would fold them up. And when I would fold them up, so within a cell, I would have this intracellular enzyme. Now, the second word was extracellular enzymes. So you see that whenever an enzyme is made within a cell, but the reactant on which the enzyme has to work, this lies outside of the cells. So the enzymes, they have to be released out. So such enzymes that are released out of the cell, so they are termed as the extracellular enzymes. And among the extracellular enzymes, I need to quote you an example in case of the human body. You see that I have got this muscular bag in my alimentary canal. So this is stomach. And in the stomach, we have got the food in the empty space. Now, in the lining of the stomach, we will have these cells because these complex particles, they can't go into the cells to be broken down. So these cells, they would release enzymes out of them. And these enzymes, now since they have come out of the cells into the empty space of the stomach, so this is an example of an extracellular enzyme. Likewise, if I were to quote you an example in case of the microorganisms, and among the microorganisms, we have got the name of the fungus and we have got the name of the bacteria. You see that if I have got this complex organic food molecule and this is the bread, and from somewhere this fungus or the bacteria, they have deposited or they have come on toward the surface of the bread. Unlike us, they don't have the teeth in them, isn't it? So if these microorganisms, they don't have the teeth, then how come they were able to digest this complex organic molecule? So the thing is very simple, that these microorganisms, they would release enzymes out of them. And when the enzymes, they would be released out of them, so these complex organic molecules that we have got, they would be broken down into the simple ones. And ultimately, those simple molecules, they would now be used up by these bacteria and the funguses. So the simple thing is that these extracellular enzymes for the second thing, now they would be released out of the body of these decomposers. And among the decomposers, we have the bacteria and we have the funguses. And these microorganisms, they would release extracellular enzymes out of them. And when they would be released out of the cell, so they would break down the complex dead organic matter into the simple substances and later they would absorb them up. Now, here I would quickly take you toward some of the technical terms that you need to have an understanding of if you have to crack this topic. So, among the first definition or the technicality that we have to understand, so this is an active site. Now, what an active site is, like if you were to see here, for this enzyme, like if I make another diagram, so if I say this is an enzyme, and within the enzyme we have got some specific shape or we have got some specific space within the enzyme molecule where the reactant molecule would come and bind with. So what we say is that this active site is actually the three-dimensional location or the position that we have got within the enzyme molecule where the substrate is gonna come and would bind with it. Okay, now moving onward, if I were to tell you about the substrate, so the substrate would be a reactant particle on which the enzymes would be working on it. Now, if you were to say that if I am trying to be denoting the enzyme molecule structure that looks like this and the substrate structure that looks like this, so you see that this substrate does not have the shape similar to the enzyme molecule, but rather than the shape of the reactant particle or the substrate is complementary to the enzyme's active site. So this substrate would come, this would bind into the enzyme's active site, and as a result of that, we are making a complex out of it. And that complex that we have made, so this would be entitled as an enzyme substrate complex, we say. Okay, so for today's class, we have seen that what enzymes are, what are the characteristics of enzymes, how many of the type of enzymes there are, and what are the technical terms. For the next class, we will see that how come the enzymes they're able to work on their substrates. So see you in the next class.